There are two main branches in our nervous system: the central nervous system, which consists of the brain and the spinal cord, and the peripheral nervous system, which consists of afferent, efferent, and interneurons. Through the cooperation of these two primary systems, these neurons within the systems allow the body's organs and organ systems to communicate and ultimately allow the organism to survive and perform other metabolic processes. For example, if a person touches a hot plate with his or her hand, the signal is first received by the sensory neurons in the hand and is then sent to the spinal cord, which finally sends a signal to the motor neurons to move the hand away immediately in order to prevent any further injury. At the same time, the sensory neuron sends a signal to the brain to sense pain. Between the sensory or motor neurons and the spinal cord or brain, there are interneurons that transfer this information from one to the other. Furthermore, there are glial cells that help support, nurture, and insulate the neurons. This system of neurons make up the nervous system, which in turn helps maintain metabolic processes, as well as allowing us to communicate with the outside world.
Here is the structure of a neuron. A typical neuron has many dendrites, which are highly branched extensions that receive signals from other neurons. The dendrites are branched off from the cell body, which contains the neuron's organelles, including the nucleus. The axon hillock is the region where the axon joins the cell body, and it's where signals are generated. The neuron has an axon, which transmits signals. Axons are wrapped by a layer of electrical insulation called myelin sheets, allowing for fast conduction. Myelin sheets are produced by oligodendrocytes in the central nervous system and Shawn cells in the peripheral nervous system. The spaces between the myelin are called the nodes of Ranvier, and the transmitted signal jumps across the axon from node to node using saltatory conduction. The inward current produced by the rising phase of the action potential at a node travels all the way to the next node, where it depolarizes the membrane and regenerates the action potential. Action potentials will be explained more later. The axon ends in branches that are called synaptic terminals, and they transmit information to other cells at the synapse. The neuron works based on channels and pumps that move sodium and potassium ions. The blue channel and dots represent sodium, and green represents potassium. The red pump is a sodium-potassium pump, and it uses ATP to actively maintain the concentration gradients of sodium and potassium because it constantly pumps 3 sodium out and 2 potassium in. This helps to maintain the resting potential of a neuron. The ion channels are selectively permeable and help to maintain a neuron's resting potential because a resting neuron has many open potassium channels. Because the concentration of potassium is much higher inside the cell, the chemical concentration gradient favors a net outflow of potassium. But since the ion channels don't allow other anions inside the cell to accompany potassium, the outflow of potassium leads to an excess of negative charge inside the cell, creating a voltage gradient. Shown here are the approximate concentrations in millimoles per liter for sodium and potassium in the fluids within and surrounding a neuron. When a neuron is at rest, the concentration of sodium is much higher outside the cell, and the concentration of potassium is much higher inside the cell. These concentration gradients are maintained by the sodium-potassium pump and represent a chemical form of potential energy. Converting the chemical potential to an electrical potential involves the ion channels mentioned earlier. As ions diffuse through the channels, they carry units of electrical charge with them. A resting neuron typically has a membrane potential between negative 60 and negative 80 millivolts, and both the concentration and voltage gradients help to generate action potentials.
At first, the neuron is at a state called resting potential. However, once the neuron is active, the membrane permeability and membrane potential start to change. As seen in this graph, the neuron at the beginning stays at a constant negative 70 MV, but once it is active, the membrane potential begins to change as Na plus channels begin to open and thus increasing the permeability of Na plus, which is called depolarization. Once there's enough Na plus within the neuron, or once the membrane potential meets the threshold, the rest of the Na plus channels start to open and creating a very positive potential on the membrane as seen in the graph. This massive inflow of Na plus from the voltage-gated sodium channels triggers a massive membrane voltage change, which is called the action potential. As seen in this graph, the voltage change or action potential is seen at the tip of the curve. Once the active action potential travels down the axon and out through the synapse terminal, the membrane potential lowers significantly through repolarization, which is due to the closing of the voltage-gated sodium channels. Once repolarization occurs, K plus channels begin to open, which then allows K plus to flow out of the neuron. This in turn changes the membrane potential to an even more negative state, which can be seen in the graph as the decrease in membrane potential. It then enters into hyperpolarization, which goes lower than the resting potential due to an outflow of positive ions or the inflow of negative ions. Finally, the neuron returns to the resting potential, or negative 70 MV, where the neuron does not send any signals after potassium channels close. Sodium channels remain inactivated during the last period, and so a second depolarizing stimulus cannot generate another action potential, and this is called the refractory period.
An action potential functions as a long-distance signal by regenerating itself as it travels from cell body to synaptic terminal, analogous to a flame traveling down a fuse. The process begins as the first action potential is generating, causing the influx of sodium ions at a location. The depolarization of the action potential then spreads to a neighboring region, starting a new action potential. The previous region loses potassium ions and repolarizes. This process is then repeated throughout the length of the axon. Once the signal reaches the synaptic terminal, neurotransmitters are released, causing changes in the postsynaptic neuron to occur. When an action potential depolarizes at the synaptic terminal membrane, which is the bulb at the end of the axon in which neurotransmitter molecules are stored and released, voltage-gated calcium channels are opened. This creates calcium ions to rush into the synaptic terminal. The increased calcium ion concentration causes the synaptic vesicles to fuse with the presynaptic membrane, or the membrane of the synaptic terminal. The synaptic vesicles are membrane sacs containing neurotransmitters at the end of axons. This causes the neurotransmitters to be released into the synaptic cleft, which is the gap between the presynaptic and postsynaptic membrane. The neurotransmitters then bind onto the receptors of ligand-gated ion channels in the postsynaptic membrane, or the membrane of the dendrite, allowing both sodium ions and calcium ions to enter. The synaptic transmission ends when the neurotransmitter is taken out of the receptor. Some neurotransmitters are inhibitory, meaning that they cause hyperpolarization in the postsynaptic membrane, making it more difficult for the postsynaptic neuron to generate an action potential. Other neurons are excitatory, meaning that they cause depolarization in the postsynaptic membrane, facilitating the generation of an action potential in the postsynaptic neuron.